And this is the outline of the talk. We're going to focus really on the non-compaction cardiomyopathy, which is, of course, a hallmark of Barth syndrome. One of the things that we have spent some time over the last year or so doing is looking at as many genes as possible implicated in non-compaction, which would include, of course, the tafazin gene. We have been doing some kinetics and recovery experiments as well with the question of if tafazin is restored, will function also be restored? And then some thoughts about potential therapeutic intervention based on really very few experiments. So in terms of genes implicated in myocardial non-compaction, about 4 or 5 years ago, Josef Finsterer in Austria put together a nice review and at the time listed 29 genes. As everybody knows, there's a lot of genetic heterogeneity as well as clinical variability with non-compaction. Here's a picture from a recent review article that was co-authored by my colleagues at NYU. And so this shows a picture which you've all seen of non-compaction. It really looks like a spongy-looking myocardium. The way I like to tell my patients is instead of your heart muscle looking like a nice sirloin steak, it looks more like a sponge. These pictures are from our mouse model, from the tafazin knockdown mouse model, published in 2012, and we had discovered that the pups, the newborn pups, as well as the embryos, had developed non-compaction with tafazin knockdown. And what we and others have observed is that there seems to be some problem with cardiomyocyte, or at least cellular proliferation. Most of the cells in the embryonic heart are cardiomyocytes, and they may be differentially affected depending on whether they are in the compact zone or the trabecular zone. And this is just a phosphohistone 3 staining with the lights is very difficult, but at a gross level, and we didn't have too many of these sections, so we didn't do any quantitation, it seems also that the proliferation is affected. And so we wanted to generate a hypothesis. This is not a hypothesis proving type of experiment, but we wanted to, there's no mechanistic understanding of why something like tafazin in an embryo, you're thinking mitochondria, you're thinking energy, why in fact should the myocardium fail to compact? Can we identify any potential therapeutic targets? And we thought, well, based on our experience, based on some of the other mouse models that have led to non-compaction, sought to link genes that are implicated in non-compaction to dysregulated cardiomyocyte proliferation. That was the hypothesis we wanted to generate. I'm not going to go through all this because it was in a poster last night that our student, Sebastian Piombo, presented, but suffice it to say, we now have collected 50 genes through a thorough scouring of the literature, and what we've also done is looked at all the literature to try and link whether or not these genes are directly related to cardiomyocyte proliferation. So the 50 genes, about 30 have been linked to human disease, 15 to 20 have been in mouse models. A few of them, I think it's about 6, were actually found in both humans and in mice, and most have either a direct or at least an indirect link to cardiomyocyte proliferation. Only one had no link, and that was, I'm blanking. DAM1, and it's felt to be sort of more of a migrational, I think, as opposed to a proliferative defect. And then in a few studies, no link was looked for, and so no link was found. Anyway, I hope you visit our poster, and we're hoping to have this published soon enough. Onward. So one of the things that we've been interested in is whether or not when you induce tafazin knockdown and induce this cardiomyopathy, can it be reversed in this mouse? The idea would be, of course, that in human patients, when you get the cardiomyopathy, when you get any cardiomyopathy, sometimes it's irreversible. You're giving therapies that may help with symptoms, that may improve function, but you want to get to the heart of the problem. And we didn't know if any of the damage was irreversible. As you know, mutations in tafazin underlie Barth syndrome, and we think that a lot of this has to do with whether or not there's enough of this mature tetralineal cardiolipin. But cardiolipin, as it turns out, this is an important question because it's been implicated in other common diseases. Barth syndrome, of course, is very rare, and people may ask, well, why study such a rare disease? Why throw so much effort into it? But in fact, hypertensive cardiomyopathy and diabetic cardiomyopathy, which are very common diseases, are also implicated when it comes to cardiolipin biology. And so if we can understand cardiolipin biology, we may be able to help, in fact, a huge number of people by understanding a rare disease. The unanswered questions in terms of tafazin knockdown in this cardiomyopathy is, can this cardiolipin deficiency and the damage that comes from it, can it be reversed? Well, can heart function be reversed? Can arrhythmias be reversed? There's probably underlying calcium homeostasis, dysfunction of that, dysregulation of that. We haven't heard much about that this session, and I don't think there's a lot of data out there, if any at all. But mitochondria and calcium are intimately linked, as it turns out. Cardiolipin profiles, can they be reversed? The mitochondrial ultrastructure. And then we've now heard a lot of stuff about reactive oxygen species. And so protein carbonylation as well as lipid peroxidation, can those injuries also be reversed? I do not claim to have very much data about most of this stuff, but I want to throw this up because, of course, this is about throwing out ideas so that we can all work together toward the common cause. And what are the kinetics of tafazin knockdown and the changes in your abnormal cardiolipin profiles? All right, so this is just to show, this is an old slide. This is, Mindong Ren actually presented these data 2 years ago, but it shows that we have efficient knockdown. I know that there's been some question about how long one should feed the doxycycline to these mice. These mice were fed for at least 3 or 4 months at least, and we have deficiency in mature cardiolipin and a great increase in the monolysocardiolipin. And the Western blot shows that the tafazin is greatly knocked down. So there's no question in our mice about this. All right, so these are the first real data that I'm going to show. And what we've done here is to essentially do a stress echo. I have heard some questions through these sessions about whether or not you can study the mice while they're stressed, running, exercising, something like that. We know that the cardiomyopathy in adult mice in which tafazin is knocked down is relatively mild. So it does not completely mimic the human condition. What we have done to try and unmask this cardiomyopathy is to give a single IP dose injection of isoproterenol. That's a beta agonist, revs up the heart, revs up the blood pressure, everything else. And what you see typically in a wild-type mouse, under resting conditions, it might do this. You give isoproterenol, and within about 2 minutes, it does this. The heart rate jumps hugely, which is here. The heart rate jumps up into the 600s, even in an anesthetized mouse. And the contractile function, as measured by the shortening fraction, is what we do, goes from something like 30 or 40% to 80, 90%. It's a huge increase. And the heart rate goes way up. So this is the heart rate response to isoproterenol. And this is what happened. We assume that we have restored tafazin. I admit to you that we did not check it because our mice were lost because of Hurricane Sandy shortly after these series of experiments. We have the samples somewhere, the tissue samples, but we have not analyzed them. But what this shows is that if you give doxycycline, you knock down tafazin, and then you stop the doxycycline, what you can do is, what happens, this is a TAS mouse, and it doesn't show much of a heart rate response at all over a 20-minute observation period. The wild-type mouse, will increase the heart rate, and then it's sustained over 20 minutes. And if you stop the doxycycline to allow recovery, you get exactly the same thing. So it seems that the heart rate response does, in fact, recover after you stop induction of knockdown. If you look at the contractile response, it's the same thing. So here at baseline, without isoproterenol, you get a milder decrease in the contractile function. This is exaggerated when you give isoproterenol. This is on doxy. And then once you stop the doxycycline for several months, the function completely normalizes. Now, we haven't done the experiments to look at things like fibrosis, look at the ultrastructure. We haven't had a chance to do that. But nevertheless, on an organ level, it seems to completely recover. All right. And this goes along with the lipidomic data that Michael Kiebisch and his group published a year or two ago, where when they stopped the doxycycline induction for a couple of months, the lipidomics went back to normal. So it seems to be in line with that. All right. So potential therapeutics. There's now, I think, emerging data over the last 2 years that reactive oxygen species may underlie the developmental cardiomyopathy that we see in our embryos, as well as the the cardiomyopathy that is seen in the adult mice. And so some of the approaches we've thought about taking would be feeding N-acetylcysteine, which, among other things, is an antioxidant, increases glutathione stores. Resveratrol, which has many effects, but can also be an antioxidant, can also stimulate mitochondrial biogenesis. Bezafibrate, which is in fact an ongoing effort by the Barth Syndrome Foundation to try and start up a clinical trial. And then a genetic cross, and we'll talk about this. This is a catalase overexpressing mouse specific for mitochondria. This is just a cartoon of reactive oxygen species and sort of how the mitochondria try and take care of this. There's a superoxide dismutase, catalase, and glutathione stores that all help to regulate levels of reactive oxygen species within the mitochondria. N-acetylcysteine comes in up over here. I recognize because we discussed this at the last conference that some of the patients are being given cysteine as supplements and stuff, and there are other possible effects. So antioxidant effect is not the only one. I think I presented these data, but not in a quantitative way. And what we had done was feed N-acetylcysteine during induction during pregnancy. And what we seem to have found was that the non-compaction cardiomyopathy was improved by feeding N-acetylcysteine. This is an approved drug. It is not given usually on a chronic basis, but is given as a single dose, for example, when patients undergo CAT scans with contrast to look at kidneys instead, to look at whatever, but it's to try and reduce the amount of renal toxicity of the contrast. And it's felt it does that because of antioxidant activity. Anyway, the amount of spongy myocardium or non-compaction seems to be less when we fed these mice N-acetylcysteine. What we want to do now, of course, is to look to see whether they actually survive, whether they can grow up, whether their cardiomyopathy is better, whether the ROS really is decreased. We haven't even shown that, and I admit that that's a big gap in our data. These data were presented 2 years ago by Mindong Ren, but it shows basically that if you take these TAS knockout mouse embryoid bodies, for whatever reason, resveratrol will, here it is, will actually reduce slightly your monolysocardiolipin peaks and increase your tetralineal cardiolipin. So based on these data, we decided, and also it seems to improve the cardiolipin profile in skin fibroblasts from Barth patients. These are all Mindong's data. And so based on that, we thought we would try and feed our mice and see if we can get any beneficial effect. We had one litter before, again, Hurricane Sandy intervened. There were 7 that were born in the litter. 1 died immediately. 1 died later. 5 made it to weaning. And 2 were TAS knockdown mice, which in our hands was very unusual. We observed pretty much universal lethality, either prenatally or very shortly postnatally. So for us to find a survivor, and in fact 2, was very unusual. Again, we don't have the data that would fill in the gaps. We don't know what their hearts look like, whatever else, because what happened was then this colony was wiped out. It was lost. We did manage to weigh them. They seemed to weigh a little bit less. Actually, there was a screw up in the cages, but it was all related to Sandy. So we didn't actually get a chance to study these mice beyond learning that at 5 weeks, we had survival of at least 50%. We are assuming that the 2 other pups that died were TAS knockdown mice. And those are only resveratrol data in vivo. The bezafibrate data, these again were presented 2 years ago. And again, feeding bezafibrate to TAS knockout cells appears to have helped with the mature cardiolipin profiles. And again, based on this, and this is just a graphic representation of the MLCL to cardiolipin ratios, seems to be helped by bezafibrate and resveratrol. And based on this, we thought, okay, we're going to feed our mice bezafibrate and see what happens. This is the heart rate response, and it looks better. Here again is a TAS knockdown mouse under normal conditions, and if you have wild type, they have a more robust heart rate response to isoproterenol stress during their echoes. And whether we fed resveratrol or bezafibrate, they all seem to have a relatively normal heart rate response. Unfortunately, we couldn't get a good contractile response to either bezafibrate or resveratrol, this kind of contractile response on echo under isopaternoral stress. So here is just at baseline, and then under isopaternoral stress, the wild-type mice really increase their contractile ability, but the under conditions of this is just regular, this is treated with bezafibrate, this is treated with resveratrol. As you can see, the contractile response was not any improved. The heart rate response was, but the contractile response was not. So whatever seems to be going on may be incomplete. Then we tried another approach, again, before Sandy intervened. The MCAT mouse is a very interesting mouse. It was developed out of the University of Washington by Peter Rabinovich and his group. And what it is it's a catalase overexpressing mouse, but specifically in the mitochondria. So there's a transgene that they introduced, but it's specific to mitochondria. And they've discovered that these MCAT mice do better if they have a cardiomyopathy, if they have some other cardiac disease that's introduced, as opposed to their PCAT mouse, which is out in the cytosol. So overexpressing catalase in the cytosol doesn't help. If you overexpress in the mitochondria, it does help. So we decided, hey, let's see what we can do if we can cross these mice with the TAS mice. So we did a number of crosses. It took a number of months. We had to finally find some mice. And then we sacrificed them at birth, and the first thing we were looking for is our non-compaction cardiomyopathy. And unfortunately, and I can't locate the slides right now, again, because of Hurricane Sandy, it still showed the non-compaction, that there was no difference in the crossed mice with both genes, transgenes, as opposed to without the MCAT. Back to that slide about mitochondria and ROS and the ROS pathways, there are other places that you can intervene, and perhaps catalase is not the place to intervene. So I'm sorry, I'm humbled by the data that have been presented at the session so far. I recognize that this is not necessarily a lot, but our take-home message is, I think, there's a strong suggestion, I think, based on our detailed analysis of what's been published, that non-compaction, and non-compaction even in Barth syndrome, may be related to cardiomyocyte proliferation and its dysregulation. And so it's not just a matter of energy. It might be energy, but I think there's a lot of data that suggests that there's enough ATP and enough energy. So it may be something else. Mitochondria are known for signaling at this point, and so it may be a signaling thing. And in fact, a lot of our genes that we uncovered are transcription factors, zinc finger proteins, and a couple of cell cycling proteins. Restoring tafazin seems to restore cardiac function, therefore the cardiomyopathy may not be reversible. I don't know if this is because of confounding factors within the mouse model, but it does have implications for gene therapy or enzyme replacement therapy. If you have a cardiomyopathy, even if it's been for a while, and in a mouse, a year is almost a lifetime, there's a potential to still reverse the cardiomyopathy. Antioxidant drugs may improve cardiomyopathy. We have to be careful about this because what we have intervened with may not be specifically antioxidant, number one. Number two, the whole antioxidant market here in the United States and around the world is, of course, a huge one, and people take a lot of antioxidants. There are commercials about antioxidants, but you have to be careful because most of those have not shown to have any beneficial effects on diseases, and it may be that you have to target the antioxidant specifically into mitochondria. So we have to be careful about the role of reactive oxygen species and how we're going to treat this. And there is some precedent. So George Porter's lab up in Rochester a couple of years ago published this paper in Developmental Cell that really provided some nice evidence that reactive oxygen species are critical to cardiomyocyte differentiation and development and proliferation. If you mess around with reactive oxygen species at different stages in the embryo, you are going to, in fact, change your time trajectory of cardiomyocyte differentiation. And so it's not just about what we think about in terms of ROS damage and whatever else. And then Bill Poo's paper that's been thoroughly discussed at this point, and the fact that mitotempo, a mitochondrially targeted antioxidant therapy, seems to rescue the cardiomyocytes from the iPSCs. And there are other papers as well. So some acknowledgments. You know how sometimes people like to say, oh, we thank you for your gift of these antibodies, or interference RNA. Well, I'd like to acknowledge our anti-collaborator, whose name is Sandy. Despite all this, we, like the families of Barth patients and the Barth patients themselves, we believe that we will still persevere and we will continue to move this forward. But on a serious note, Mindong Ren, Michael Schlama, Young, and David Stokes from structural biology have been my collaborators, and we would not be able to do this without the entire team. We've had some students. Asha Mahajan previously has worked on some of the recovery experiments, and currently Sebastian Piombo is in the audience. Lee Neshawa couldn't make it. He's a medical student. He's been helping us with some of this as well, and funding especially from the Barth Syndrome Foundation. Thank you very much.
Okay, we have some time for questions.
Yeah, that's very kind of inspirational. But I wanted to ask you, so that your antioxidant interventions were all essentially non-specific, except for this CAT, mitochondrial targeted catalase, and with which you seemingly didn't have any success, and you concluded that hydrogen peroxide is not the site. Well, if we're talking about Barth and if we're talking about Ross and Barth syndrome, so then the oxidation target will be cardiolipin. And I wanted to remind all of us now, so that in 1984, Fulvio Ursini in Italy has discovered glutathione peroxidase 4, which is a mitochondrial glutathione peroxidase, which utilizes phospholipid hydroperoxides as its substrates. And so GPX4 will reduce not hydrogen peroxide, but glutathione phospholipid hydroperoxides, including cardiolipin hydroperoxides, and stop this oxidative chain of events. So if I would cross anything with TAZ knockout, that was GPX4 overexpressor, not catalase. Okay.
Okay.
That's my point one. And my point two, do we have time for long points? And my point two is that kind of as I was thinking and looking at Michael Schlama mostly and maturation of cardiolipins, so, and if we also think about oxidative stress and ROS, so then there may be signals that selectively are formed from oxidation of tetralinoleal cardiolipin. And these signals actually are known to be 9 and 13 hydroxylinoleic acid. And I'm asking everybody and you, so whether you know anything about these 2 hydroxylinoleic acid homologues as potential regulators or on their involvement in Barth syndrome. No?
Yeah, I don't know anything. I think too
because they have a different kind of anti-inflammatory, pro-inflammatory, as many lipid mediators. But these 9,13 linoleic acid hydroxy are powerful regulator of many functions.
I think too that the whole story of the involvement of reactive oxygen species during development is a hugely incomplete story. I mean, it's already incomplete when we're looking at damage in the adult heart, but it's even the gaping holes in knowledge during development especially. Michael.
Great talk, Colin. I just have a very practical question. So when you withdraw doxycycline, how long does it take for the tafazin level to come back to normal? And then do you really need to wait 3 to 4 months before you see normalization of the heart rate and the contractile response?
Yeah, excellent question. We're in the midst of doing those experiments. What we want to do is logically, right? So your mRNA is going to change first, then your proteins, then your lipids, then your organ level physiology. So we have to take all of that into account, and then we also want to look at electron microscopy. So we don't have enough time points right now. The original paper that Zaza published in JBC in 2011 suggested after a month off doxycycline, your tafazin levels were down by about 45%. Michael Kiebish's work suggests that the kinetics may be a little bit faster because that was a lipidomics paper, and after 2 months, everything seemed to recover to normal. I think there's also some stuff about the mouse, how you induce it, how you feed it, what you do with it, that will also influence the kinetics of all this stuff.
I've been very intrigued by both your studies and Bill's with the idea of the reactive oxygen species, which are known to be, that will regulate many different pathways. As a dysmorphologist, I'll point out, and I presented on this in the past, that there's clearly an increased instance of malformations, congenital malformations in Barth syndrome that don't hold together very well, but it's clearly very increased. And the idea that either the reactive oxygen species or the lipid abnormalities are affecting tissue-tissue or cell-cell interaction, I think may be a fundamental part of the syndrome. And whether it's reactive oxygen species or lipid or both, I think again, as a dysmorphologist, I see the heart as a dysmorphologic heart.
I think we agree the heart is dysmorphologic here.
Okay, okay, we do have two questions.
So in your heart rate response experiment, I was wondering, do you expect the beta-adrenergic receptor signal was affected in your knockdown?
I'm sorry.
I was wondering whether the beta-AR signaling is affected.
Yeah, absolutely a fair question, because I seem to remember too, now that you raised the question, that mitochondria, in heart failure can be linked even to beta-adrenergic signaling and stuff like that. So I don't know that, the answer to your question. We have not looked at that, but that's a very good question, whether or not there's changes in the receptors that may explain all this.
First, just a quick comment about this recovery. When you're right, when we got first data, it was 45% recovery, but after this we checked several times, and it seems the data is not always consistent. In some cases we have a 45, but in some cases we have almost 90% is still down after 1 month of taking of the doxycycline. And that's, I blame, because it's a food, doxycycline is introduced as in the food, in the rodent show, and in your model, I think it's much better. It's with the water.
It's in the
right?
water, yeah.
I think it's more plastic, and it's more reliable. And then another quick question: have you seen any signs of non-compaction in the, like a 2-3-year-old
In
older mice? Because
No,
those data.
no. And I'm wondering too if in this model, so, our speculation is that we see what we see because the dose during gestation is much higher than what you or Barry introduced. And is there a sweet spot that will allow us to see some non-compaction, but not so bad that there will be survival, and that they'll survive into adulthood with a more severe cardiomyopathy than you're otherwise seeing? I don't know if there's that sweet spot. I don't know that any of us are working on that. That's just a bunch of cages with different doses. So it's kind of a boring, if necessary, experiment.
Okay, thank you.
Thank you.